Hey everyone, this is Dr. Marcon, and I will be doing the discussion on Lab 22, red blood cells, as well as blood typing. So in lecture, we went over general characteristics of red blood cells. Again, in the bone marrow, committed progenitor cells will differentiate into large nucleated erythroblasts. As these cells mature, the nucleus will condense and the cells will shrink in diameter. Now, in the last stage before maturation, the nucleus is pinched off and phagocytized by bone marrow macrophages, while other organelles like mitochondria break down and disappear. The immature reticulocyte will leave the bone marrow and enter the circulation, and this reticulocyte will then mature into a erythrocyte in about 24 hours. So here we see the stem cell maturing in the presence of erythropoietin into a reticulocyte before becoming a red blood cell within 24 hours once the reticulocyte has been released into the circulation. And while that happens, the organelles are broken down and will eventually disappear. So, general characteristics of our red blood cells, we know that the shape of red blood cells are biconcave discs, which help increase surface area, as well as speed up diffusion of the respiratory gases oxygen and carbon dioxide. We know that these cells are flexible and have the ability to squeeze through narrow vessels. Also, another general characteristic is that they are anucleate, meaning there's no nucleus or organelles. And because they have no mitochondria, they cannot carry out aerobic metabolism, but they do have enzymes for anaerobic metabolism and, again, get their primary source of ATP through glycolysis. Before a red blood cell matures into an erythrocyte, it is in the reticulocyte stage, and this is an immature red blood cell with a nucleus. And again, the major function of our erythrocytes is to carry gases such as oxygen as well as carbon dioxide. So the first part of the lab, we're doing a total red blood cell count using a hemocytometer. Hemo means blood, cyto means cell, meter means counter. We counted 5 squares, 1 through 5. We can see in these corners 1, 2, 3, 4, 5, and each square had 4 by 4 squares within it. So we counted these 5 squares. We multiplied by 10,000, which adjusts for dilution and volume counted. We know that it is influenced by dilution accuracy and sample homogeneity. So then we wanted to get the total number of red blood cells, and this is in millions per microliter. Normal number is approximately 5 million red blood cells per microliter. And we know that red blood cell count is usually a little bit higher in men than women. For this lab, we also obtained hematocrit numbers. Hematocrit being packed red blood cell volume, and which is the fraction of the total sample of blood. And we used a microcapillary reader to measure the hematocrit. So the formula for hematocrit is packed red blood cell volume divided by total blood volume, and this will give us a percentage. So hematocrit is given in a percentage or plasma percentage, and you can calculate the other. So we know that whatever the plasma percentage is, whatever the hematocrit percentage is, this will equal 100%. And usually hematocrit is about 42% of the blood sample. Now, for hematocrit, of course, men will have a higher hematocrit level, whereas women will have a lower hematocrit level. And we use this number to help us diagnose conditions such as anemia. And if we have a hematocrit percentage that is less than 37, we can suspect that the patient has anemia. Hemoglobin is the main component of red blood cells. It is a very important protein. Its structure is a tetramer, meaning it has 2 alpha and 2 beta subunits, and each subunit contains a heme or iron molecule, and each heme binds to a molecule of oxygen. So hemoglobin is an important protein found in RBCs. We know that there are about 280 million hemoglobin molecules per red blood cell. And when we take a hemoglobin concentration number, the number is usually about 14 grams of hemoglobin per deciliter of blood. But imagine 280 million hemoglobin molecules per one red blood cell, and we know that a normal human has about 5 million red blood cells. So again, the hemoglobin is a tetramer having 2 alpha usually and 2 beta subunits, and there are 4 hemes attached to an iron molecule per hemoglobin, and each hemoglobin molecule binds to 4 oxygens, because we can see we have 4 hemes that will bind to oxygen. The function of hemoglobin, of course, acting as a carrier protein to allow transport of oxygen as well as carbon dioxide. And we talked about hemoglobin also being important as being a buffer for blood, and we talked about that during lecture. We know that hemoglobin does carry several gases, and depending on the gas, we can kind of subcategorize the type of hemoglobin. For example, oxyhemoglobin. This is when a heme molecule binds to oxygen. Now we know that iron in the presence of oxygen becomes oxidized and creates a red color. That's why oxyhemoglobin bound to oxygen shows up as red within the arteries. So arteries have oxyhemoglobin, or a high oxygen content, whereas deoxyhemoglobin basically means that there is a reduced number or no oxygen bound to the iron, and this will cause that bluish or cyan color within the veins. And then carbaminohemoglobin, this is when the carbon dioxide is carried on an amino acid. Now carboxyhemoglobin, this is when we have heme bound to carbon monoxide, and we know that carbon monoxide has a higher affinity for hemoglobin compared to oxygen, so carbon monoxide acts as a competitive inhibitor of oxygen. It has greater than 200 times a higher affinity for hemoglobin. That's why carbon monoxide is very serious. And carbon monoxide poisoning is treated by giving high amounts of oxygen. So to kind of get an idea of the affinity of carbon monoxide for hemoglobin, for example, if we had 50 hemoglobin proteins, this means there are 200 sites for binding of either oxygen or carbon monoxide, because 50 times 4 is 200. Out of the 200 sites, 199 of those sites would bind to carbon monoxide, while only 1 oxygen would bind to hemoglobin. That's how much of an affinity carbon monoxide has for hemoglobin. The way that we sort of measured hemoglobin concentration, we used the Tallquist method with comparing bond paper color. So we had a drop of blood go onto the bond paper, let it dry, and then we compared the color of the drop of blood to the colors that we found on this card. And we wanted to closely match the color on the card with the color of the drop of blood as closely as possible to give us that hemoglobin concentration. Now, a normal hemoglobin concentration is about 14 grams per deciliter. Of course, men would have a higher hemoglobin concentration compared to women. Now, if we have a hemoglobin concentration less than 12, we would consider pathological condition of anemia. Note also that there is a specific ratio of hematocrit to hemoglobin concentration, and that ratio is 3:1. Now, if we know this ratio, we can sort of estimate the other. So, for example, if we know that hemoglobin has a concentration of, say, 12, we can estimate if we multiply 12 by 3 that the hematocrit would be 36. So this is a really good ratio to know, especially when you're trying to get an idea of hematocrit percentage as well as hemoglobin concentration. Now, mean corpuscular volume measures the cell size of the erythrocyte, and this is important, especially if we want to see whether or not there is some sort of pathology involved. So MCV means mean corpuscular volume. This is the average size of the cell, and we get the MCV with this formula. So we have the total RBC volume divided by the number of red blood cells. So in this formula, we see that hematocrit times 10. So, and then divide that by the red blood cell count, which is in millions. So, for example, if we have a normal hematocrit of 42, we multiply that by 10, and then we divide that by 5 to get the MCV of 84. Now, normal mean corpuscular volume is between 82 to 92. So, if a MCV is below 82, we would consider that cell small. So it would be considered microcytic. Normocytic, so within the normal range, and then macrocytic above 92, so being a very large cell. And we have different anemias that would cause a macrocytic anemia. Mean corpuscular hemoglobin concentration is a measure of hemoglobin concentration. So to get the MCHC, we would use this formula. So we would take the hemoglobin concentration, multiply it by 100, and then divide it by hematocrit. So for example, if we had a patient with a hemoglobin concentration of 14, we multiply that by 100 and divide that by hematocrit, which normally would be about 42. So our MCHC, or mean corpuscular hemoglobin concentration, would equal 33.3. So again, this is the hemoglobin concentration within the red blood cell. And also we can see whether or not it is within normal based on the color. So we know that hemoglobin concentration affects the color of the cell. The more hemoglobin, the darker the cell. So if we have, for example, an MCHC within normal range, so that is between 32 to 36, we consider the erythrocyte normochromic, so a normal shade of red. However, if it is below the normal range, so below 32, you would have a much paler cell. So we would consider this cell hypochromic, so below normal color. If we had a cell above the normal range, so above 36, we could see a darker erythrocyte, so this would be considered hyperchromic. In lecture, we talked about how oxygen carrying capacity of the blood can be determined by several factors. So these factors include total red blood cell counts. We know there are approximately 5 million red blood cells per microliter or per cubic millimeter of blood. Another factor includes hematocrit concentration. So hematocrit percentage normal is about 42%. We would consider anemia if that hematocrit percentage went below 37%. And again, here is the formula for hematocrit. You would take the packed red blood cell volume and divide that by the total blood volume to get the percentage. And we know that the plasma percentage plus hematocrit would give you 100% of this sample of blood. Hemoglobin concentration, again, we're saying that normal hemoglobin concentration is approximately 14 grams per deciliter, or 280 million hemoglobins per red blood cell. And we would consider anemia if the hemoglobin concentration fell below 12 grams per deciliter. So patients with anemia have a reduced oxygen carrying capacity. I kind of went over this in lecture, but again, anemia, basically you have a compromised ability to carry oxygen, and an indicator of anemia includes a reduced total red blood cell count, a decrease in the hemoglobin concentration to possibly 12 grams or below, and a reduced hematocrit percentage. We're saying that less than 32% is considered, we can also consider anemia. So causes of anemia could be due to a reduced red blood cell number. This is usually due to an increased destruction, also possibly a decreased production of red blood cells. Also, we can have insufficient hemoglobin, meaning too little hemoglobin, or an inadequate hemoglobin. This could be due to a quality problem. So, for example, there could be an underlying pathology such as sickle cell anemia, which changes the shape of the red blood cell, not allowing for proper binding of the maximum amount of oxygen to hemoglobin due to a change in the shape. So with reduced red blood cell number, red blood cells could be normal in size, and hemoglobin concentration could. With anemia, there could be a reduced number of red blood cells. Now, the red blood cells may be normal in size, and normal hemoglobin concentration just have less. So this could be due to a decreased production of red blood cells. This could be due to dietary deficiencies such as iron, vitamin B12. We talked about how if we don't have intrinsic factor, we can't have the absorption of vitamin B12, which is important for red blood cell production, as well as a deficiency of folic acid. Other conditions that could cause a decreased production of red blood cells include bone marrow disease, where progenitor cells that turn into red blood cells could be decreased. Also, an inadequate amount of erythropoietin. Reduced red blood cell number could also be due to an increased destruction or an increased loss of red blood cells. So if we have, for example, weakened red blood cells, such as red blood cells that are formed due to hereditary spherocytosis causing an abnormal shape of the cell, these cells could be easily destroyed by organs such as the spleen. Or if we have an increased blood loss, such as monthly blood loss due to menstruation or even a bleeding peptic ulcer, this could also reduce the number of red blood cells. Another reason that we could have anemia could be an insufficient concentration of hemoglobin, and this could be due to iron deficiency. Iron deficiency being one of the more common causes of anemia. Another cause of anemia could be inadequate hemoglobin. An example would be sickle cell anemia, which also transforms the shape of the red blood cell, causing a sickle cell shape of the red blood cell, and this, of course, affecting the amount of surface area where oxygen can bind to the cell. We characterize anemia by the cell size as well as color. So when we talk about cell size, we use the suffix cytic. And usually cell size or any deviations from normal cell size is a DNA-based problem. So if we have a cell that is microcytic, this is a cell that is considered too small. If we have a cell size that is normocytic, we consider this just right. And if we have a cell size that is macrocytic, macro meaning big, this is a cell that is, of course, too big. If we have a deficiency due to hemoglobin amount, we characterize the cell by its color and use the suffix chromic. So usually hemoglobin amount is an RNA-based problem because we know that RNA is responsible for the production of protein, and hemoglobin is a protein. So for example, if there's too little hemoglobin, a cell would be considered hypochromic, meaning it is below normal cell color. Normal amounts of hemoglobin produce a normochromic cell, and too much hemoglobin will produce a darker cell, so we consider the cell to be hyperchromic. So again, anemias are described using a combination of red blood cell size as well as color. So don't forget to go over these different formulas that we use to calculate both mean corpuscular volume, MCV. This is the average cell size. And then we use mean corpuscular hemoglobin concentration to calculate the average hemoglobin amount within the cell. So we use this formula, mean corpuscular volume. We take the hematocrit percentage, multiply that by 10, and divide that by the absolute number for the red blood cell count in millions. So for example, if we have 5 million red blood cells, we just use the number 5. And for MCHC, we take the hemoglobin concentration, multiply that by 100, and divide that by the hematocrit percentage. So, for example, if normal hematocrit, we'll use the number 12 just because it's an easy number, and then we multiply that by 100 and then divide that by whatever the percentage of hematocrit is. So knowing the normal ranges for MCV as well as mean corpuscular hemoglobin concentration, we can then determine the size and color of the cell. And given these numbers, we could also identify the type of anemia if anemia is present. So for example, we know that the normal mean corpuscular volume is between 82 and 92. So if we see that we are given that MCV is 79.8, it is below the normal range, so we would consider that the cell would be microcytic, so small for its cell size. And then mean corpuscular hemoglobin concentration, the normal range for that is 32 to 36. So if we are given that the MCHC is below 32, it would be considered having too little hemoglobin, so it would be hypochromic. So if we are given these numbers, we would classify this as a microcytic hypochromic anemia. So make sure you are aware of the normal ranges for MCV as well as MCHC, so that if you are asked, you can help identify the type of anemia if anemia is present. Iron deficiency is the most prevalent cause of anemia. Usually this type of anemia is a microcytic hypochromic, so cells of iron deficiency anemia are small and they are very pale in color. This is the most prevalent anemia, and usually it's due to chronic infection. So at this point, with chronic infection, the body can sequester iron in the liver to help fight infection. However, iron deficiency anemia is usually due to nutritional deficiencies. And we can see the percentages of iron deficiency anemia and the prevalence throughout the different groups. So with women, about iron deficiency anemia affects about 20%, 3% in men, 20% in children, and we can see that the larger population is pregnant women. And this is due to an increased blood volume. If there's a large plasma increase due to pregnancy, it is difficult to increase the red blood cell output to match the increasing blood volume. Other common anemias includes macrocytic normochromic anemias. Usually this is pernicious anemia, and this is usually due to a vitamin B12 deficiency, especially because of the lack of intrinsic factor being very important for the absorption of vitamin D12. So now that we've gone over general characteristics of red blood cells, and how we measure certain components of red blood cells, such as hematocrit, total red blood cell count, as well as hemoglobin concentration, we then get into blood typing. Blood typing is actually pretty fun. So red blood cells have on their surface specific antigens. Antigen is actually a shortened term for antibody generator. So these substances stimulate an immune response or antibody formation. So cell surface molecules such as proteins and carbohydrates give red blood cells a unique identity. So we also have genes that determine blood type antigens. With our ABO blood typing, we have type A, type B, or type AB, or an O type. And then we consider the Rh antigen, or the Rh factor. It's actually Rh because of Rhesus monkeys. Rhesus monkeys were used to determine this antigen. And you can be either Rh positive, having that Rh antigen, or Rhd antigen, or you can be negative, meaning you don't have that antigen. So our blood type is actually determined by what antigen is on the surface of the red blood cell. So for example, if we have type A blood, that means the A antigen is present on the surface of the red blood cell. If a patient has type B blood, that means the B antigen is on the surface of the red blood cell. If a patient has type AB blood, that means both A and B antigens are on the surface of the red blood cell. If a patient has type O blood, that means there are no antigens on the surface of the red blood cells. So again, it's these surface antigens that determine blood type. So this is just another picture showing the different antigens that are on the surface of the red blood cell that help determine your ABO blood type. Again, if there are no AB antigens on the surface of the red blood cell, that means you are type O. If you have the type A antigens, you are type A. Type B antigens, you are type B. And then if you have both AB antigens on the surface, then you are type AB. Also is the same for the Rh factor. If you do have that Rh antigen on the surface, then you're positive. If you do not, you are negative. So the human body has 23 chromosomes, and we know that there are genes that determine blood type antigens. We can use, if you see this picture here, this is actually a human karyotype. A karyotype is just a map of chromosomes. So we have one chromosome coming from mom and one set comes from dad. So we know that specific chromosomes actually determine blood types. So for ABO, the genes for ABO, with regards to the antigens that are on the surface of the red blood cell, these are found on chromosome 9. So they actually have mapped out the different chromosomes. So we can count 1, 2, 3, 4, 5, 6, 7, 8, 9. So it's these genes that determine your blood type. And then whether you are Rh positive or negative, that gene is found on chromosome 1. So humans have 2 copies of each gene, again, one coming from mom, one coming from dad. And we consider the genetic makeup of a person their genotype. So genotype being the genetic makeup of the genes that they receive from each parent. And so it just says which 2 versions of a gene you have. Again, one coming from mom, one coming from dad. So a single locus determines your ABO blood type. Think of locus sort of the address. The address of where the gene is located, again, is called the locus. The version of the gene is called an allele. So, for example, allele A will have the A protein, allele B will have the B protein, and allele O will have the O protein. So the phenotype is basically the outward expression of the genetic makeup or the genotype of a person. So the phenotype, for example, would be a person's ABO blood type. And here we see that there are different genotypes that can produce the same phenotype. So for example, if you have type A blood, you can either have a genotype of A allele coming from mom, A allele coming from dad, or the genotype AO. Okay, so one allele coming from mom, one allele coming from dad. For type B, the same. You can either have 2 Bs, so a B, or you could have a B and an O genotype to make the type B blood type. For AB, you have to have 1A, 1B, and that will give you the blood type AB. And in order to have the O blood type, you have to have both O's for your genotype. And we know that blood type is actually inherited from our parents. We get 1 gene from each parent. So for example, if we have a father whose blood type is A, and we know that their genotype is AO. And then dad has kids with mom whose blood type is B, and her genotype is BO. Not body odor, but, her genotype being BO. We have children with many different blood types depending on the combination of these alleles. So, for example, one combination, we could have a child with AO blood type, and we have another child that could have AB blood type, another that could have type B blood but have a BO genotype, and then we could have one combination where the child is type O. And we can actually use Mendelian genetics with Punnett squares to determine the different blood types or blood type combinations that can occur. So here on this next slide, we see the Punnett square made famous by the monk Gregor Mendel. When I went to Villanova, we actually had our science lecture halls was named after Mendel. So we would go to Mendel Hall for our science classes. So here we see the genotypes of both parents. Mom is type A, but she has the genotype AO, and dad is type B, but he has the genotype BO. So again, we use a Punnett square to kind of find out the different combinations of blood types, and we see that 1/4 or 25%, there's a 25% the child might have type AB blood, and we have 25% that the child could have type O blood, and then another 25%, or 1/4, 1 out of the 4, that the child could have type B, but have, what we call heterozygous dominant for type B, and here type A blood being heterozygous for type A. Now, if we mix two parents that have type AB blood, we can see other combinations that can occur. So 1/4, 1 out of 4, so 25% of the child having type A blood, but being homozygous for type A, so having 2A genes or 2A alleles, and then a 25% of the child being homozygous for type B, so having 2 B alleles for type B. And then we have 2 chances, so 2 out of the 4 being a 50% chance that the child might have a combination of type AB blood. So we use this Punnett square to help us determine the percentages for the combinations that can occur between two parents and knowing their blood types. We actually had to practice doing Punnett squares. I remember taking a genetics class, and that was usually super fun. You can also actually use the Punnett square to determine paternity, whether or not a certain blood type can occur from parents knowing what their blood types are. And do a whole Maury Povich and say, you are not the father because you are this type blood, and we know your parents' blood types. So that's kind of fun. So plasma antibodies dictate red blood cell surface proteins. If, for example, we have a person who is type A. So type A means they have the A antigens on the surface of their red blood cells. Now, our bodies are so made in that if we have an A antigen, we have in our plasma B antibodies, so anti-B antibodies. Because if we had anti-A antibodies in our plasma, being type A blood, these antibodies would attack the red blood cells, causing an immune response. So we know that plasma antibodies do not recognize red blood cell antigens of that type. So, for example, if we have type A, our anti-B antibodies in the plasma will not recognize these antigens. Okay, so we have no agglutination. Our antibodies are not going to be attacking our own red blood cells. That just would not be compatible for life. So if we have, for example, if we are type B blood, we have B antigens on the surface of our red blood cells, meaning we have anti-A or A antibodies within the plasma. Now note for patients with blood type AB, meaning they have both A and B antigens on the surface of the red blood cells. If we have both A and B antigens, that means there are no antibodies in the plasma. Okay, so there are no antibodies that could attack the red blood cells. Because there are no antibodies within the plasma of patients or people with type AB blood, we consider these people to be universal recipients, meaning they can accept either type A or type B blood because there are no antibodies in their plasma that would cause an immune reaction to blood that is donated to them, being either type A or type B. So type AB people are known as universal recipients. Let's look at type O blood. So type O blood, they have no antigens on the surface of their red blood cells. Okay, so no antigens. However, that means they have both A and B antibodies within their plasma. Okay, so they have both anti-A anti-B within their plasma, patients with type O blood are known as universal donors. Okay, so they can donate their blood to anybody. And that is because they have no antigens on the surface of their red blood cells that can cause an immune response or agglutination. Agglutination is just a fancy name for clumping of cells. So here we just see characteristics of the ABO blood groups. Again, type O blood, there are no A or B antigens on the surface of the red blood cell. However, within the plasma, there is both anti-A and anti-B antibodies. For type A, the red blood cell has A antigens on the surface of the red blood cell, and they will have anti-B within the plasma, meaning they can only receive type A blood, because if they receive type B blood, their anti-B antibodies will cause an immune response with the type B blood if it is given or transfused. That is why it is very important to make sure that before any transfusion of blood that you do the appropriate blood typing and matching. And you want to match the blood as closely to the recipient as possible, because we do not want to have agglutination. Agglutination can actually cause some pretty serious damage. So for type B blood, there are B antigens on the surface of the red blood cell and anti-A antibodies within the plasma, meaning they cannot receive any blood that is type A, because if they receive type A, the anti-A antibodies will bind to that blood and cause agglutination and wreak havoc. Now for type AB blood, they have both A and B antigens on the surface of their red blood cells, and note there are no antibodies within the plasma, so no anti-A or anti-B, so they can actually receive any blood, so they are universal recipients. So type A or type O being the universal donors, type AB being the universal recipients. So again, this is just the table of the different types of blood, possible genotypes, genotype combinations that create the blood types, as well as the antigens that are present on the surface of these red blood cells, as well as the presence of the antibodies within the plasma. So when red blood cells with group A antigens on their membranes are mixed with plasma containing antibodies to group A, the antibodies cause the blood cells to clump. So agglutination is just a fancy term for clumping. So when we mix blood types, we increase the risk of agglutination, clumping together of the red blood cells, and this can block small blood vessels. This is a lot different from clotting. These large molecules will block small blood vessels and decrease the amount of blood that can flow to that tissue. So here we see donor with type B blood. They have anti-A within their plasma. And we have a recipient with type A blood with B antibodies within their plasma. So as we can see, when the type B is donated to a patient with type A blood, their antibodies are going to cause agglutination. So blood cells from the donor will agglutinate to the antibodies of the recipient. This can cause damage to red blood cells, so usually red blood cells can burst. Side effects include a disruption of kidney function, or these large molecules could cause clumping, which blocks blood flow in the capillaries, and then this compromises oxygen and nutrient supply to cells as well as tissues. So again, it is very important that we do blood typing, blood matching before transfusing a patient, and make sure you're transfusing the proper blood type to the recipient. So here we see the antibodies agglutinating with the red blood cell with the antigen on its surface and causing these large molecules. Again, agglutination of the red blood cells with the antibodies of the recipient can block small blood vessels, and usually these cells become damaged or lyse, and again have some major serious effects on the kidneys. So agglutination can cause red blood cell destruction, blockage of small blood vessels, and will reduce oxygen and nutrient supply to tissues. Safe transfusion does require the proper matching blood type. And again, we talked about a universal recipient being type AB blood because they have no antibodies within their plasma. And then the universal donor is type O blood because there are no antigens on the surface of these red blood cells to cause agglutination within the recipient. And we have different incidences seen as percentages with regards to the different types of blood. So we can see that type A within the US, it has an incidence of about 42%. Type B, about 10%. AB is 4%, and type O is 44%. Now that percentage of incidence can actually vary with different ethnicities and different racial groups. For example, I know Filipinos, for example, there's a higher incidence of type B, specifically type B positive among Filipinos. I, for example, am B positive, and that makes sense because usually I'm a pretty positive person. But and Asians actually do think that there is a correlation between blood type and personality. I don't know how much that is true, but I know for me, I'm definitely type B positive. And make sure you get your blood type done. It's an important thing to know, especially if you're working in the healthcare field. If you want to donate blood, or God forbid you're in some sort of accident where you have to be transfused blood, it is very important to make sure you know what your blood type is. So it is very safe. It's very important to match blood type. We want to make sure that there is no agglutination that occurs between the donor and the recipient, because antibodies in the donor plasma can cause mild agglutination. So here we see that type O can be donated universally to any blood type, and type AB can receive any blood type because of the lack of antibodies within the plasma. Rh antigen is just another cell surface protein. We were able to find this antigen through working with rhesus monkeys. The Rh address or locus is on chromosome 1, and basically you're either positive or negative for the Rh factor. So if both parents are positive, you'll be Rh positive. If one parent is positive, you'll be heterozygous for Rh positive. If both parents are negative, then more than likely you'll be Rh negative. So incidence for Rh factor or the Rh antigen to be Rh positive, it occurs mostly in 85 to 90% of patients. Rh negative, about 10 to 15. And here we can see percentages. If we have, for example, two parents that are heterozygous for being Rh positive, the majority of them, about 75% of their offspring will be Rh positive, whereas you'll have one 125% chance of the offspring being Rh negative due to the combination of alleles. If we actually put this in a Punnett square, we can determine that 75% will be Rh positive, 1 bit will be homozygous positive, whereas 50% will be heterozygous positive, and then we'll have that 25% chance of being homozygous negative, because you have to have both parents being negative, or both alleles being negative, in order to produce an Rh negative offspring. So Rh antigen is a cell surface protein. If you are Rh positive, we do have a harmless anti-Rh or antibody to Rh. However, when we're talking about hemolytic disease of the newborn, if, for example, mom is Rh negative, however, her husband or the father of her baby is Rh positive, they conceive a child and the baby is Rh positive. The first pregnancy usually is harmless, no real complications. However, once the mother is exposed to an Rh positive baby, she will later produce antibodies against the Rh protein or Rh antigen. So any subsequent pregnancies, these antibodies will then attack the developing fetus and causes what we call hemolytic disease of the newborn. So hemolytic disease of the newborn, also known as erythroblastosis fetalis, Rh positive active antibodies can be generated by fetal mother blood exchange or blood transfusion. So active anti-Rh positive antibodies from the mother will attack the Rh positive fetus and causes jaundice. Basically increased destruction of red blood cells. Bile is produced, or bile is found, or I'm sorry, bilirubin is found in the blood, causing the jaundice within the newborn. So that is hemolytic disease of the newborn. Now, this can be prevented if we give the mother Rhogam. Rhogam is a medication. Basically, it's an injection that we give to the mother with antibodies that will destroy any fetal Rh positive cells. So here is just a lovely table showing you the different blood types and the different antibodies that are present. So for example, if the red blood cell, if the blood type is A positive, that means we have the type A antigen as well as the Rh antigen on the surface of the red blood cell. So if the patient is A positive, that means they will have B antibodies within their plasma. If, for example, the patient is A negative, meaning they do not have the antigen for the Rh factor. So again, they will have the A antigens on the surface of the red blood cells and B antibody within the plasma. So if the patient is negative, meaning they do not have the Rh antigen on the surface of the red blood cell, they will have the antibody, the Rh antibody postnatally within the plasma. And so far, so if patient is type B positive, they will have the B antigen on the surface as well as the Rh antigen on the surface. So they will have the anti-A antibody in the plasma, but no Rh antibody within the plasma because of the presence of the Rh antigen on the surface of the red blood cell. And here we see a patient who is A negative. Actually, that is a mistake. It should be B negative. So I'm going to write a letter B here, so you guys can make that correction. This should actually be B negative, not A negative. So if the patient is B negative, meaning they have the B antigen on the surface of the red blood cell, oops, let's write that again. So B negative here, I don't know why I erased that. So B negative here, meaning they have the B antigen on the surface of the red blood cell. However, they do not have that Rh antigen. So within the serum, they'll have the A or anti-A antibodies as well as the Rh antibodies within the plasma. AB positive, so both A and B antigens on the surface as well as the Rh antigen. So that means they have no antibodies within their plasma. So again, these patients can be universal recipients. They can receive any blood. Here we see that the patient is AB negative, meaning they do not have the Rh factor. So they will have the Rh antibody post-natally. Patient with O, type O blood, no antigens on the surface of the red blood cell. However, if they're positive, they have the Rh antigen. So no antigens, meaning they have both anti-A and anti-B in the plasma. Again, these are usually type O is a universal donor. And O negative having no Rh antigen on the surface. So they will have Rh antibodies within their plasma, as well as type anti-A and anti-B antibodies within the plasma. So that is blood typing in a nutshell. Hopefully you guys are able to see the patterns with regards to, blood type being determined by the antigens on the surface, as well as what antibodies would be present if we have specific antigens on the surface of the red blood cells, and what could happen if we incorrectly blood type a patient and give them the incorrect type of blood when we transfuse them. And this could cause some serious consequences.